All right, so this little screencast is going to be on bacterial transformation, and it ties into this lab that we're trying to make bacteria grow. So, first big idea, bacteria increase in variability in offspring through transferring plasmids from one to another. There's other ways that we'll talk about, especially through mutations and stuff, that bacteria can have variation. But this whole idea of plasmids, you have this single longer round chromosomal DNA within a prokaryote or a bacteria, but then they also have this other smaller ring plasmid DNA. So this plasmids can also be picked up from the surroundings, which we're going to talk about with this lab. And variation is a prerequisite for natural selection. If you don't have variety, you can't have different ones being selected for, if otherwise they'd all get wiped out the same. So transferring plasmids with genes that code for proteins that block or inhibit antibiotic chemicals is the basis for antibiotic resistance. And we're going to do a little bit of that in this lab too, which is pretty cool. And humans can also transform bacteria with unique recombinant plasmids. So we're going to deal with this P-glow plasmid um, and try and make bacteria glow basically under UV light. So the second big idea here is that transcription is regulated. Operons, and again, there's a couple of operons within eukaryotes, but we typically think of operons within bacteria. So there's promoter sequence of DNA, operator sequence, and then a bunch of genes that follow that. So a regulatory protein can often sit on the operator, like switch, basically to turn it on and off. And if certain environmental presence of particular chemicals are around, you can actually turn on genes um, if they're there, or otherwise it can stay turned off. Okay, so this lab, basically this is what we're looking at. We've got an E. coli type of bacteria, Escherichia coli. It's got its chromosomal DNA, big loop. And then what we're going to try and do is take our special recombinant plasmid and get the E. coli to take it up into its cell. And then it's going to express not only its genes on its chromosome, but it would express the genes within this recombinant plasmid. But this lab has a couple of unique things to it because in order to make this E. coli receptive to picking up the plasmid, we're going to mix the E. coli along with plasmid separately. You don't have these together. You don't buy it like this. You get them separate. You put them in, and then ultimately you try to transform this bacteria. And to make it receptive, you actually heat shock it. So you get it really cold, and then you get it really hot for a very specific amount of time, and then you put it back on the cold, and that process of heat shocking these E. coli make them more receptive to picking up the plasmids from the environment. And some of that will be successful, but the success rate is really specific to doing the lab correctly. Because if you don't heat shock it, only like 10% of the plasmids will be taken up, or 10% of what could be possibly taken up. If you do it for like 90 seconds, you're only going to get half as many as what you would get if you did it for 50 seconds. So it's this specialized way to get the E. coli receptive to picking up plasmids from the environment. So this plasmid, it's pretty special here. The way this works, you have a gene here that basically is on. Okay, so this gene right here is going to be making enzymes that are resistant to antibiotic, and the antibiotic we're specifically going to be resistant to is ampicillin. Now, this GFP is actually a gene from a jellyfish, and it's a green fluorescent protein. And the way this has been engineered is that only if this gene gets turned on will this gene get turned on. And the only way this gene gets turned on is if arabinose sugar is present in the nutrient medium. So if there is the right sugar, then it'll make the protein to make them glow. If the sugar is not there, it won't make it glow. One way to think about this is getting back into this whole lac operon. And the lac operon, again, it's a promoter sequence of DNA, an operator sequence of DNA, and then a whole bunch of genes, and then a repressor protein is sitting on the operator. As long as that repressor protein is sitting on the operator, RNA polymerase cannot go and transcribe these genes. The only way the repressor gets off of the operator is if lactose is present. So in order to make the genes that help break down lactose, lactose actually has to be in the environment. So once lactose is around the bacteria, the lactose will land on the repressor, take it off, and then the RNA polymerase can transcribe the genes to ultimately make the enzymes to break down the lactose. It's genius. So, one way of thinking about this lab is that we can genetically engineer this and we can put the green fluorescent protein or green fluorescent um, gene for a green fluorescent protein into this sequence. So then if the sugar is present, not only will it make the genes to break down the sugar, it'll also, because we've engineered it that way, also end up making the proteins to make it glow. So as we do this, we go through this process of heat shocking, trying to get the E. coli to pick up the recombinant plasmid, and we have it in this little test tube here. And then we take this from the test tube and we streak it along an agar plate, and we incubate that and see if it'll end up growing. Now, one thing to keep in mind, I already said that this whole heat shocking process is kind of difficult to get the right conditions to get the E. coli to accept as many plasmids as possible. Even if you do it well, not every single E. coli within this is going to pick up the plasmids. So you're still going to have a whole bunch of E. coli that are not transformed. So you are hopefully getting as many transformed bacteria as possible, but even in this heat shocking process, you're still going to have quite a few that did not end up picking up the plasmid. So let's think about this for a sec. And this is ultimately kind of some results we're looking at. So we don't just transform the E. coli, we're also going to have basically a control group. We're going to have a group that did not get the plasmid, and those are going to be our wild type E. coli. And then we're going to try and take these wild types and grow them on different types of agar, and we're going to take our transformed E. coli with this new glowing and antibiotic ampicillin resistant plasmid and try and grow it on some different things. So right here, what this is showing is that if you grow regular E. coli on just a nutrient agar, this Loria broth, you're going to get a lot of bacteria growth, and you can kind of see that there. Now, if you took this normal E. coli and grew it on a nutrient agar plate that also had ampicillin, which is an antibiotic, you're going to get nothing to grow. Everything's going to get killed because this wild type E. coli gets destroyed by ampicillin antibiotic. Now, with our transformed E. coli here, we take this and we culture it on this broth, this nutrient agar that also contains ampicillin, and we are going to get some that survive. because this thing is making the ampicillin protein, ampicillin resistant protein inside of it. Now, why doesn't this look like this? And the big reason is this right here is um, all of these, all of them growing. This down here is going to have some of these that transferred, but it's also going to contain some of these ones that did not pick up the plasmid. So everything that did not pick up the plasmid is going to die, and it's not going to be fully grown like this. But all of these ones that are recombinant that picked it up will survive. Now, as we go into this last set right here, these did not glow because in order to trigger the green fluorescent protein, arabinose sugar has to be present. So now in the culture that you're trying to grow this stuff on, if you have the ampicillin um, antibiotic, you're still going to kill anything that did not pick up the plasmid, but now because arabinose is present here, now the ones that do survive that have been transformed are also going to make protein within them that cause them to glow under UV light. Okay, so let's take a look at how this is actually assessed often. So this says, in a transformation experiment, a sample of E. coli bacteria was mixed with a plasmid containing the gene for resistance to the antibiotic ampicillin. Plasmid was not added to a second sample. Samples were plated on nutrient agar plates, some of which were supplemented with antibiotic ampicillin. The results of the E. coli growth are summarized below. The shaded area represents extensive growth of bacteria. Dots represent individual colonies of bacteria. So basically what we're looking at here, you have these that were not transformed. You grow wild type with no ampicillin, you get a whole bunch of growth. You grow wild type on ampicillin, it's all going to die. You grow these that have been transformed on no ampicillin, they're all going to live. You grow these on ampicillin and you have not nearly as much growth. Why wouldn't you have as much growth? Because not every single one will be transformed. So if we're taking a look at this, plates that have only ampicillin resistant bacteria growing include which of the following? Okay, I'm going to give you a second to try and think about this. Which ones only contain ampicillin resistant bacteria? And hopefully you said 4. Okay, so which of the following best explains why there is no growth on plate 2? Correct answer is that the initial wild type is not ampicillin resistant. It did not have a plasmid within it to protect it from ampicillin. All right, moving along. Plates 1 and 2, I'm up here. Plates 1 and 2 were included in the experimental design in order to. Why in the world did they even do this wild type E. coli? What is the purpose of doing that? The correct answer is to make sure the E. coli cultures are viable. Okay, next one. Which of the following statements best explains why there are fewer colonies on plate 4 than on plate 3? I've already kind of gone over this. Hopefully you guys said B, because not all of them are successfully transformed. In a second experiment, the plasmid contained the gene for human insulin as well as ampicillin resistance gene. Which of the following plates would have the highest percentage of bacteria that are expected to produce insulin? and it is 4. These that survived not only are going to be ampicillin resistant, but then they would also be able to make insulin. So you know for sure that these are actually making insulin. You don't know how many are actually in those other ones. And none would be here because they don't pick up the plasmid at all. All right, so now we're looking at this arabinose operon. You have regulatory sequences, you have genes. You have a promoter, you have an operator, and then you have different types of genes here. So the regulatory sequences of the operon controlling arabinose metabolism, ARA operon, were studied to determine whether bacteria can respond to changes in nutrient availability. It is predicted that if those regulatory sequences are functioning properly, the bacteria will produce the enzymes involved in arabinose metabolism, structural genes B, A, and D, in the presence of arabinose. If a gene that encodes a green fluorescent protein is substituted for the structural genes of the operon, activation of the regulatory sequences can be assayed by GFP expression. So basically they're saying, like, if you can tell if they glow, then they must be turning on, then arabidose must turn on those genes. It must be an operon. And culture of E. coli cells underwent a transformation procedure with a plasmid containing the regulatory sequences of the ARA operon directly upstream of the gene encoding the GFP. This is very similar to what I was just showing you guys with the lac operon. The plasmid also confers ampicillin resistance to bacteria. Samples were then plated on different types of culture media. Note, the GFP fluoresces only under UV light, not under white light. And then we're going to have a table of the results. So one thing to keep in mind is that the ampicillin resistance is not within this operon. Only the GFP is within this operon. The ampicillin resistance is separate from it. So you have this culture media. You have agar in all of them. This positive indicates that the substance is present, and the minus says it's not there. So in this first one, there's no ampicillin, there's no arabinose. You grow the stuff on there, they are all white. Next one, you have agar, you have ampicillin, but you do not have arabinose. They are all white. They the colonies grow because you transferred ampicillin resistance. This next one, there's no ampicillin, but there is arabinose. Under white light, they're all white. They only grow, they would only be green under UV light, but we have some green, but mostly are white here. Now, in this situation, you have ampicillin and you have arabinose, and they are all green. They're all green because anything that would be white here would have not picked up the plasmid. So you have some white in this because you're also going to have some bacteria growing that have not picked up the plasmid. In this situation, only the ones that pick up the plasmid are going to survive because they would be ampicillin resistant, and then they would also glow green. All right, so the question here, which of the following, I mean, that's a heck of a lot of information so far just to answer one multiple choice, but which of the following can best be used to justify why the GFP is expressed by E. coli cells after transformation with the plasmid? Why is the GFP expressed after transformation? Everything that we read, all of that kind of stuff, comes down to the presence of arabinose in the agar activated the expression of genes located downstream of the operon regulatory sequences. So the sugar triggered the making of the new genetically engineered GFP that got put into the plasmid. And that's that. So that is the end. Hopefully you guys learned something about bacterial transformation.